Hello, this is the last part, the fourth part to presentation on cells. In this part, we'll talk about transcription translation. Transcription is the process of copying a strand of DNA into a corresponding strand of messenger RNA, and it's also known as the messenger mRNA or messenger RNA. So this is going to be just a single strand of messenger RNA, and the difference is that with DNA you have two strands, and that those two strands together cannot fit through the nuclear membrane. It can't get out of the nucleus. So DNA has to transcribe itself into a new form called messenger RNA. So during transcription, you're going to have transcription happens in the nucleus. You should know that. That's a major thing to know, and that nucleotides are going to be ordered in a complementary manner. So a cytosine, which is represented by a C in DNA, will dictate a guanine, or shown by a capital G in messenger RNA. Likewise, a G in DNA dictates a C in messenger RNA. So G to C, C to G. A thymine, represented by a capital T in DNA, dictates an adenine, a capital A, in messenger RNA. However, here is where it gets a bit tricky. An A in DNA dictates a U, not a T, as we might expect in messenger RNA. So when you're going from DNA to RNA, if you have an adenine, it's going to dictate a uracil be installed on the messenger RNA strand. So the following DNA sequence would dictate the corresponding messenger and RNA sequence: A to U, T to A, G to C, C to G, A to U, T to A. New mRNA strand will then move out of the nucleus via the nuclear pores and to the ribosome to form protein synthesis. This is where translation takes place. The process by which a mRNA strand is decoded into a protein. Most sets of 3 consecutive bases code for a specific amino acid, which is the building block of protein. These are referred to as codons, and a few, however, are reserved as stop and start codons. These signal the ribosome to initiate the protein synthesis or cease it. At the ribosome, the mRNA strand is translated into a protein. This is accompanied by yet another strand of RNA being formed to arrive us back at our original DNA sequence. This new strand of RNA is called tRNA, or transfer RNA. It follows the same base pairing rules that we follow going from DNA to mRNA. So here are U will go back to an A goes to U, C goes to G, G goes to C, U goes to A, and A goes to U in the tRNA strand. At this point, we translate the tRNA into a protein using the codon rules. A codon table is needed, so you'll find one attached to your notes, or you can find one by searching the web. All right, that concludes our chapter and our topic on cells. Please watch all 4 videos as many times as you need. You can use them for any assignment, remember, and unless otherwise notified. Thanks.